I started out as a researcher about 20 years ago as a graduate student studying hearing loss in an avian model that was bred for low frequency song. And the model was very intriguing because birds normally have the ability to regenerate their hair cells after damage. But this type of bird was unable to regenerate its hair cells properly and regain normal hearing. My lab at Hopkins, however, uses mostly mouse models of age related and noise induced hearing problems with a little bit of work on genetic hearing loss. My lab studies how the brainstem is affected by different forms of hearing loss and how the feedback pathways projecting from the brain to the ear play a role in hearing deficits. My lab is especially interested in linking structural changes in the auditory system to behavioral and perceptual deficits like hearing noise difficulties or hypersensitivity to loud sounds and tinnitus. We also sometimes do comparative studies using interesting species because my background is in comparative hearing. So we're very interested in species that are exceptionally resistant to aging and hearing loss. We're primarily interested in understanding basic mechanisms of hearing loss, as well as potential strategies to protect against degeneration of the auditory pathways and to improve hearing. Rubenstein funding has allowed us to pursue novel research questions about the projections from the brain to the ear, which we call the auditory efferent or oligocochlear system. Normally, the system helps to protect against hearing loss, and it facilitates hearing and noise by adjusting the gain in the cochlea through a feedback loop, sort of like the way a thermostat in a house works. Weakening of these pathways can lead to problems with sound perception, but we're also interested in what circumstances strengthen these pathways. So we've had a really unique opportunity to look at the strengthening of these pathways through the Rubenstein funding at Johns Hopkins. Johns Hopkins is a really great place to be investigating this pathway because we have some of the world's foremost experts on faculty. And we also have excellent opportunities to collaborate with other labs. We've been working with a bat lab to study specializations in the auditory efferent system. So bats are really interesting for us because they live much longer than other small mammals, and they're presumed to be resistant to hearing loss throughout their lifespan. And they rely on a keen sense of hearing to survive because they need echolocation to forage for food and to navigate through the world. But a bat that can't hear in the wild won't likely survive for long. What's really unusual is that in nature, they're exposed to hours and hours of very loud sounds from their own vocalizations and from other bats. Yet they don't seem to develop hearing loss. These kinds of sounds would cause loss of hearing loss in humans and other species. So we believe that they have evolved efferent specializations to protect against this age-related and noise-induced hearing loss. But those mechanisms are really not very well understood in bats. There are only a few studies from decades ago. Our collaboration with the Bat Lab investigates this enhanced protection against hearing loss. The species we work with seem to have enhanced auditory efferent systems with more cells and larger synapses in the ear. And we think that those increased number of cells and the larger synapses actually allow for better protection after noise exposure and against age-related hearing loss. And those specializations might also help the bats filter out irrelevant sounds so they can pay attention to the important sounds that they need for foraging and flight. Interestingly, we have found that some individuals of one of the species we look at, the big brown bat, actually are deaf. This species is native to Maryland. It's a very common bat around here. And usually we work with wild-caught members of the species. But we were able to obtain some captive-bred individuals of this species, and it turned out some of them couldn't hear at all. They were completely deaf. They were missing most of the sensory cells and neurons in their ears. We think that deafness is actually related to inbreeding and other stressors that are increasing the susceptibility to deafness and possibly to deficits in the auditory efferent system. We plan to perform measurements to assess the status of the auditory efferents in young, very young bats and aging bats, see if this noise resistance and these specializations are present at birth or if they develop over a course of having exposure to loud sounds early in life. The reason we want to look at that is because our recent work with the Glovatsky lab at Johns Hopkins has shown that one branch of that auditory efferent system, the lateral efferents, are very plastic depending on acoustic exposure. And by plastic, I mean that certain proteins are upregulated in response to loud sound exposures. We might be able to identify certain conditions that allow us to boost the beneficial effects of the efferent system, um, and maybe improve hearing noise or protect against hearing loss. The idea would be hopefully that one day we can apply what we learn from animal models to human ears. So there's still a lot we don't know about the efferent system. It's not a very well understood system, even though it's been studied for many decades, because it's a very complicated system. We want to know things like what spectrum of conditions will result in this plasticity, and is the plasticity diminished with age? Meaning, is it possible that this brain to ear pathway isn't able to compensate for changing sound conditions, changing sound exposures with age? Our previous work with the Fuchs lab at Johns Hopkins indicates that the synapses of these auditory efferent neurons actually move around in the ear with age-related hearing loss. We don't really know quite what the effects are of that on hearing loss, but we think it could either exacerbate the hearing loss by turning down the cochlear activity even more, or it might actually form some sort of compensatory strategy for protecting sort of what's left in the aging ear, the neurons in the aging ear that haven't degenerated. We also don't know how well the efferent neurons are working in cells in the ear when the cells are regenerated after damage. We don't even really know if they get wired up properly after the regeneration. So the next phase of the research funding will support us in investigating these kinds of exciting questions. That's really important because it's very difficult to get funding from the NIH or other sources for exploratory types of questions and forming new collaborations. To get funding from the NIH, you really have to show the experiments are going to work out exactly the way you think they will. And often in studying the system, that's not how things happen. So I'm certain that the experiments that we're going to do are going to lead to really surprising and interesting discoveries and additional paths to pursue in the future.